Class, we have discussed about these detectors and the ideal characteristics of a detector for gas chromatograph. So today we'll proceed further with the detectors that you have in your syllabus. You have mainly four detectors. Thermal conductivity detector. I mentioned these names last class. So thermal conductivity detector, or TCD, electron capture detector. Then you have flame ionization detector, and you also have nitrogen phosphorus detector. Okay. So in short, I have given you here what each one of this actually how it responds. So thermal conductivity detector measures the decrease in the thermal conductivity of the carrier gas resulting from non-conductive analyte molecules. What it means is the carrier gas has its own thermal conductivity, and when any component that is getting eluted out comes along with the carrier gas to the detector, then the detector will sense the change in the thermal conductivity of the carrier gas or the gas that is reaching the detector. This decrease will be measured as a peak in the chromatogram. We will see this, of course, in detail. The second one is an electron capture detector. So this measures the decrease in the detector current which occurs when an analyte molecule that tends to capture electrons appears in the gas stream. This is a selective detector. So only those compounds or analytes that can capture electrons, when they come into the detector, then this detector will start sensing it. Otherwise, it does not show any um, response. Just a minute. The flame ionization detector, or the FID, last class I told you these are standard short forms, so you can be asked a question with this short form itself. So remember the short forms. Measures the increase in electrical signal as a result of ionization of analyte molecules by the action of the detector flame. This means that whenever a component gets eluted and comes along with the carrier gas to the detector, there will be an increase in the electrical signal. How it happens, we'll see in detail later. And lastly, you have the nitrogen phosphorus detector, which is the NPD detector. It's also called the thermionic detector, I told you last class, and the short form is TID. This measures the increase in current resulting from combustion of nitrogen and phosphorus containing compounds in the detector flame. So whenever you have nitrogen and phosphorus containing compounds, then you have a sudden increase in the current. This is just a short introduction to all these four detectors that you have in your syllabus. Particularly, the first three are important. I've included the fourth because it's a slight modification of the FID. These three are particularly mentioned in your syllabus. So TCD, or thermal conductivity detector, these are a comparison of the detectors that we are going to study. This is selective for all organic compounds. That means it is a general purpose detector. It is not a selective detector. It responds to all organic compounds. Its sensitivity is, however, little low. So maximum you can say 5 to 20 nanograms only. And the temperature limit, however, is very high. So it can withstand temperatures up to 450 degrees centigrade. And that's one of its major advantages. The other is any type of compound, most of the organic compounds, particularly, which is what our pharmaceuticals will be, most of these compounds can be estimated by thermal conductivity detector. The next one, electron capture detector, or ECD. Is a very selective detector. It's not a general purpose detector, it's a selective detector. It is selective towards halogens, nitrates, and conjugated carbonyls. Or, in other words, it is selective to those compounds having electronegative functional groups. Then its sensitivity is quite high. It is zero point 0.1 to 100 nanograms. So you see the lower limit is much lower compared to the TCD, and that's why we say this is a much more sensitive detector. The temperature limit is, however, only up to 350 degrees centigrade, much lower than the TCD. So those compounds which require operation temperature to be more than 350, you cannot analyze with an ECD. Then you have the FID. This is also considered a general purpose detector for all organic compounds. That means compounds with CH bonds. So that is in general organic compounds. So it is very sensitive. So compared to this TCD, you can see the sensitivity is much high. It's also zero point 0.1 nanogram. TCD doesn't go to such low concentrations. Such low concentrations can also be estimated by FID. Temperature limit is also reasonably high, 400. This is the most commonly used because of its very high sensitivity. TCD is also used because it's a general purpose detector and temperature limit is high. Coming to the last one, NPD, it is a selective detector. It is selective towards compounds containing nitrogen and phosphorus. To such compounds, the sensitivity is extremely high. You can see it's much higher than these two also. 0.001 nanogram to zero point zero 0.01 nanogram. But the temperature limit is quite restricted. And this is useful only for the nitrogen and phosphorus containing compounds. It's less commonly used in pharmaceuticals, more in the pesticide industry. So these are some other detectors. I thought I would share with you this because it's not only those four which are used in gas chromatographs. These are other detectors also. We have flame ionization, FID, I already told you, thermal conductivity, TCD, electron capture, ECD. You can see it is applicable to what type of compounds, flame ionization to hydrocarbons, thermal conductivity to all compounds, and electron capture to halogenated compounds, particularly or in general to it is selective towards all electronegative functional group compounds. Then you can also have mass spectrometer as a detector with GC. Of course, it gives rise to the hyphenated technique of GCMS, very useful because mass spectrometer is a highly sensitive and accurate, lot of advantages using this detector, but this is not the conventional GC. So you have a mass spectrometer combined with a GC instrument where the mass spectrometer acts as the detector, GC is for separation purpose. This is tunable for any species, so you can detect any species of any molecular weight and very high sensitivity. You can see it is down to the picogram level. Picogram level, what is a picogram? Comparison to gram, 10 raised to minus 12 gram is a picogram. This is Fg femtogram, so it is 10 raised to minus 15. Then you have the thermionic, that is nothing but the NPD detector. It is selective towards nitrogen and phosphorus containing compounds. Sensitivity to phosphorus is much higher than to nitrogen, about 10 times more. So if it is zero point 0.1 picogram, then for nitrogen it's 1 picogram. Apart from the four and mass, you also have electrolytic conductivity detector. Compounds containing halogen, sulfur, or nitrogen can be estimated with this. You can see these are all very sensitive detectors. Photoionization is another. Compounds ionized by UV radiation. Such compounds can be detected with a photoionization detector. 2 picogram is the for carbon. containing compounds. Then Fourier transform IR, FTIR can also be used as a detector along with GC. That gives rise to another hyphenated technique, that is GCIR. This is useful for all organic compounds. All organic compounds give a response with FTIR. So here also quite a high sensitivity, zero point 0.2 to 40 nanograms. So these are many other detectors, but in your syllabus you have only the first three. So we will be concentrating on that, and I will just give you a little brief explanation of the thermionic also. So we start with the first one, thermal conductivity detector, very important for you in your syllabus. So it's a very early detector for gas chromatography, one of the first ones to be used and still being used. The one that still finds wide application, it's based upon changes in the thermal conductivity of the gas stream brought about by the presence of analyte molecules. So what is important here is thermal conductivity of the gas stream. So um, here you need to see what are the changes in thermal conductivity of the gas stream whenever a substance gets eluted. The sensing element of TCD is an electrically heated element whose temperature at constant electrical power depends upon the thermal conductivity of the surrounding gas. I will explain a little more about this once we see the actual diagram of this. So the TCD has a sensing element which is electrically heated. Its temperature at constant electrical power depends upon the thermal conductivity of surrounding gas. The heated element may be fine platinum, gold, or tungsten wire, or it could be a semi-conducting thermistor. Just a minute. So the advantage of the thermal conductivity detector is its simplicity, its large linear dynamic range. That means over a wide concentration range, it responds accurately. 10 raised to 5 means 1 lakh. So it may be responding, say, from 1 milligram to 1 lakh milligram, a wide range of concentrations. So its general response to both organic and inorganic species, so it can respond to any type of compound, and it is non-destructive in character, which permits collection of solutes after detection. So these are the advantages. Most of the advantages are there. Only thing, its sensitivity is not as high as the ionization detectors that we'll see later on. Thermal conductivity detector actually has a number of advantages. That is why it is still used, but it has a little limited sensitivity. That is, it's limited to 10 raised to minus 8 gram solute per ml of carrier gas. If the amount of solute is less than this, then it may go undetected by a thermal conductivity detector. You can see the word here, catharometer. Catharometer is another name for thermal conductivity detector. So you could be asked, explain the construction and working of a catharometer. You need to write about thermal conductivity detector. You could be asked, explain the construction and working of TCD. That again, same answer. You have to explain about thermal conductivity detector. So it's another name of TCD. Other detectors exceed this sensitivity by factors as large as 10 raised to 4 to 10 raised to 7. That's what I was telling you. The sensitivity of the ionization detectors is much higher compared to TCD. Now I'll explain the working. Um, Try to understand this. If there is any, if you're not understanding, please ask me at the end of the. I hope you can hear me. So please, this is very important. That's why I want you to ask me any doubts regarding the working. So it consists, this is a thermostated metal block. Okay. In this metal block, two passageways are drilled. These are the two passageways. These two should be identical. Its width, its length, everything should be identical. Each passageway has fitted into it a wire or a filament. This is the filament, or it could be a wire. It is made from a metal with a high temperature coefficient of resistance, such as platinum or tungsten. What it means, this high temperature coefficient of resistance means when the temperature changes slightly, the resistance changes by a bigger magnitude, by a larger magnitude. So easily, even a small change in temperature should be easily recognized by this detector, and it will respond to it. That's why we use metals such as platinum or tungsten, which have high temperature coefficient of resistance, to make these filaments. Now, these two are identical filaments. The two filaments are connected into a Wheatstone bridge. I hope you remember a Wheatstone bridge. Wheatstone bridge has two arms, and we will be seeing the current flowing between the two arms. I'll show you next picture. Pure carrier gas is made to flow through one passageway. See, this is the reference stream. So here, this is where it is connected to the column. So the from here only carrier gas will be passing, and from here this is connected to the column. This is direct reference gas. So only carrier gas without passing through the column will be passing through this reference cell. So through this passageway, only pure carrier gas always will be passing, whereas through this sample cell or sample filament will be exposed to what is coming out of the column, or the column effluent will be passing. Sometimes when there is no elution taking place, then it will be only carrier gas passing through here also. Sometimes it will be a sample that is getting eluted along with the carrier gas. Okay. Now, what is the principle of working of the TCD? The rate of heat loss from a heated wire placed in a gas stream depends on, amongst other things, the thermal conductivity of the gas. I hope you understood this. You have a heated wire or filament which is electrically heated. What is the rate of heat loss from that heated wire will depend upon a number of things, one of which is thermal conductivity of the gas. So we will be having two identical filaments. Both are heated to the same temperature. So the rate of heat loss must be the same, provided the thermal conductivity of the gas in the cell is the same. This is the principle. Whenever there is a change in the thermal conductivity of the gas in either the reference or the sample cell, then the rate of heat loss will change for that filament. Now, if the composition of the gas stream changes, and then what will happen? Then its thermal conductivity will change. When will the composition of gas stream change? When any component from the sample is getting eluted in the gas stream. If it is only carrier gas, then the gas stream composition remains same. Same in the filament as in the same in the reference cell, same in the sample cell. Whenever a component gets eluted in the sample cell, the composition of the gas stream will change. If the composition changes, the thermal conductivity of the gas in the sample cell will change. If the thermal conductivity changes, then rate of heat loss from the heated wire will also change. If that changes, then the temperature of the heated wire will change. If the temperature changes, then the electrical resistance of the wire will be changing. If the electrical resistance of the wire changes, then you will see that the Wheatstone in the Wheatstone bridge into which it is connected, it will go out of balance. Now here you see the Wheatstone bridge. You have the two filaments, they are placed in a Wheatstone bridge. You can see here analytical cell and reference cell. The filament from the reference cell is connected here. The filament from the analytical cell is connected here. I'll just go back to this. This is the filament. So this analytical filament is connected here, and the reference filament is connected here. This is one arm of the Wheatstone bridge. This is the other arm of the Wheatstone bridge. Now, these two arms, if they are balanced, that means these are known resistances. So, known resistance and reference cell, this makes up the complete resistance of this arm, right arm, and known resistance and analytical cell resistance makes up the left arm resistance. When these two resistances are balanced, then there is no current flowing between these two. So here you have the galvanometer, which will show zero deflection. Whenever the bridge goes out of balance because of different resistance there between these two, then there will be a deflection. So now any change we were seeing when there is only carrier gas flowing, there is no elution of any component, then you have carrier gas in both these two filaments. Rate of heat loss will be the same because thermal conductivity of the gas is the same. So rate of heat loss is same. So temperature is same. Resistance is same. The bridge remains balanced and therefore no deflection. Any change in the composition of the column effluent due to elution of a component leads to a change in its thermal conductivity, which throws the bridge out of balance. So, uh, whenever you see that there is some elution or the composition of the column effluent is changing, you will see the change only in this cell. So the composition of the gas stream in this cell changes, the filament rate of heat loss in here only changes. So the temperature of this filament changes, its resistance changes. So the overall resistance of this arm now is different from this arm. So we say these two arms are not balanced. If it is not balanced, a current flows between the arms and it is evidenced by this showing a deflection. So what should be done? You have to change the resistance here till the overall resistance of this arm matches with this. How much resistance has been changed? That is the change in the filament resistance. Based on that, we can find out what is the change in temperature, and based on that, we can find out what is the rate of heat loss change that is taking place, and hence what was the change in thermal conductivity. And from that, we can find out what is the concentration of the component that was getting eluted. This is the way this whole TCD works. The out of balance potential will be related to the concentration of the component in the effluent stream. So whenever there is a change, a deflection, there is a current flowing, you will immediately see a peak appearing in your recorder. So you have these other potentiometers. Here in this diagram, you can see potentiometer. This is used to control the heating current in the filaments. Because I told you in the beginning, these two filaments are electrically heated. So that's done with this potentiometer. This potentiometer is used to attenuate the signal. Suppose the signal is too high, then it controls it. This is called the sensitivity control one. Too high signals means it will actually lower the sensitivity, so the signal will also lower. If it is showing a very small signal, it will increase the sensitivity and therefore you get a much better signal. This is how the changes are done. We come to the next of the detectors, that is flame ionization detector. This is also a very important detector from your exam point of view. So how does this work and what is its construction? What is it made up of? Before that, a general introduction. The flame ionization detector is the most widely used and generally applicable detector for gas chromatography. Why? Because of its number of advantages, which we'll see. The effluent from the column here is mixed with hydrogen and air and then ignited electrically. Most organic compounds, when pyrolyzed at the temperature of hydrogen air flame, produce ions and electrons that can conduct electricity through the flame. This is just a brief introduction. So whenever you have organic compounds, when they come into a hydrogen flame, they will ionize, giving ions and electrons. This increases the electrical conductivity of the flame, which gives a peak in the chromatogram. So a potential of a few hundred volts is applied. The resulting current is generally in the range of 10 raised to minus 12 amperes. This can be easily measured. It can be amplified also. The flame ionization detector exhibits a high sensitivity, 10 raised to minus 13 gram per second, large linear response range 10 raised to 7, and low noise. You can see the linear response is much greater than TCD, which was 10 raised to 5. Here it is 10 raised to 7. So a much higher concentration range over a wider concentration range, it gives a linear response. I hope you understand what's a linear response. If 1 nanogram, say 1 nanogram per ml concentration gives 1 microampere current, then 3 nanogram will give you 3 microampere current. That's called a linear response. A disadvantage of the flame ionization detector is that it is destructive of sample. So whatever sample is passing through a flame ionization detector cannot be collected and used for any further studies because it ionizes it changes the nature of the sample. This is the one of the desirable, so it does not satisfy one of the desirable characteristics of gas chromatograph, but otherwise it has very high sensitivity. It is a rugged detector. It can be used for all organic compounds. So these are a number of advantages which make it a very popular detector. This is the construction. I'll explain here. So this is the anode. You have wire anodes like this dipping in. When you are drawing this, please see that these anodes are drawn well beyond the tip of the flame. Your anode should not stop at this point above the flame. It should be extending beyond the flame. So this is the anode. So it will be connected to the positive terminal of a battery. This flame head or the burner head, this will be the negative terminal. So it will be connected, it is the cathode, it is connected to the negative terminal of an external power source. So here you have positive terminal, this is negative terminal. Now we come to the bottom here. Here you have the column connected at this end, and then whatever is coming out from the column, that's the column effluent, enters through this capillary. So here it comes into the detector. Just before reaching the detector, hydrogen also is combined. When hydrogen is combined and hydrogen is introduced, you will get the hydrogen flame. Here you can see air is also there. So when hydrogen burns in presence of air, it gives you the hydrogen flame. Hydrogen flame has thermal energy which is sufficient to ionize any carbon containing compound. And that's how you will see that carbon containing compounds will ionize in a hydrogen flame. So whenever component, carbon containing or organic component gets eluted and it comes to the hydrogen flame, it undergoes ionization. Ionization means it gets one electron at least gets removed from the component. So the component forms a cation and an electron is formed. The cation, being positively charged, gets attracted towards the cathode, and the electron gets attracted towards the anode. And you have to have sufficient potential between these two to ensure that the two do not combine. The positive ions should come and get attracted towards the cathode, negative should move away, that is, the electron should move away. This movement of charges sets up a current, and that's how a current is recorded. And whenever there is an increase in current because of the elution of an organic component, you will get a peak in your chromatogram. So that's how the whole thing works. Next, you have the thermionic detector. This, I think it's not mentioned in your syllabus, but since it's a modification of FID, I thought I will just give you a brief explanation of this. This is selective towards organic compounds containing phosphorus and nitrogen. Its response to phosphorus atom is approximately 10 times greater than to nitrogen atom, and 10 raised to 4 to 10 raised to 6 times larger than to a carbon atom. So the thermionic detector also responds to carbon atoms, that means carbon containing compounds, but the sensitivity is very low compared to its sensitivity towards nitrogen or phosphorus. For phosphorus, it's very sensitive, 10 times less to nitrogen and much lesser by this magnitude to carbon atoms. Compared with the flame ionization detector, the thermionic detector is approximately 500 times more sensitive to phosphorus containing compounds and 50 times more sensitive to nitrogen bearing species. These properties make thermionic detection particularly useful for detecting and determining the many phosphorus containing pesticides. That's why it's more used in the pesticide industry compared to pharmaceutical industry. And how does it become so sensitive towards phosphorus and nitrogen containing compounds? Now, as I showed you in this diagram, here you have a hydrogen flame. The modification in the NPD is above the hydrogen flame, there is a rubidium silicate bead. The rubidium silicate bead will be electrically heated. So when it is electrically heated, um, the temperature that is present, the electrical heating results in thermal energy being generated here. That thermal energy is sufficient to ionize phosphorus and nitrogen containing compounds. Carbon containing compounds actually ionize here. Its sensitivity is quite low, but nitrogen, so if you have very small amount of sample coming in, the carbon ionization goes undetected. It is the ionization that takes place at the rubidium, heated rubidium silicate bead, which actually causes the current to flow. So that's the small modification that is done in the TID. Another important detector is the electron capture detector. This is a very selective detector in its response, being highly sensitive to molecules containing electronegative functional groups such as halogens, peroxides, quinones, and nitro groups. However, this detector is insensitive to functional groups such as amines, alcohols, and hydrocarbons. An important application of the electron capture detector has been for the detection and determination of chlorinated insecticides. This is, of course, in the insecticide industry. It is also useful in the pharmaceutical industry. So this is a picture of the electron capture detector. Here you have this is the detector, and here from the column, exit end of the column, the effluent will come through. Here you have a radioactive beta emitter. So this will be emitting, this could be tritium or 63 nickel. So it can be used as the radioactive source. This will emit beta radiation, or beta electrons are emitted. So your column effluent comes here, that is carrier gas alone or carrier gas with eluted component. You have the two electrodes, the tube through which the column effluent comes in, that is made the cathode, and the wall of this chamber is made the anode. So you have the two electrodes also, and this will be the positive and the negative electrode. So whatever ionization takes place, the negative ions will move towards the positive, and the positive ions will move towards the negative electrode, setting up a current. This is how it will work. We'll see the important points. ECD detects ions exiting from the gas chromatographic column by the anode electrode. So you have tritium or 63 nickel as the radioactive source. It emits beta particles or beta electrons. Negative charges they are. The ionization, how it takes place for this detector, nitrogen is a very suitable carrier gas. So we are taking this, can also use argon. So if nitrogen is used, then what happens? Nitrogen with beta electrons gets ionized, giving you nitrogen cation, and one electron is removed from nitrogen. How the cation is formed? When one electron gets removed. And that's how you have two electrons, one the bombarding beta electron and one getting removed from nitrogen. That's the two electrons. You have nitrogen cation, which is positively charged, and the two electrons which are negatively charged. These together establish a high baseline. Why? Because these are charged species. You have sufficient potential between the cathode and anode such that they do not recombine. And especially since the electron is a very small entity, it moves away fast from the nitrogen cation. It will get attracted very fast towards the anode. So these electrons move towards the anode, nitrogen cation moves towards the cathode. This will give you a high current and hence a high baseline in the chromatogram. Next instance is when a sample is getting eluted out. So a sample which is capable of, say, a halogenated sample, capable of capturing electrons, is coming along with carrier gas into the detector. What happens then is this will capture the beta electrons. Because it's capable of capturing electrons, the beta electrons are immediately captured, resulting in X minus, an ion of the sample. Now you have nitrogen ions and the beta electron is captured, so you have X minus also. Now these two are result in almost the same number of charges, so the current should be the same, but in practice this doesn't happen because of the recombination. So X minus combines with nitrogen ion, giving neutral X and neutral nitrogen molecule. Why this happens when it is not, recombination is not taking place earlier, when there is no component, only carrier gas? That's because the electron is a small entity and it can move very fast in a given applied potential. It can move very fast towards the anode. So it avoids recombination with nitrogen cation. But now you are applying the same potential, and this is a bigger molecule. This is a bigger molecule, so it does not move, and so naturally a bigger anion. It does not move so fast away from nitrogen cation. There is sufficient time for this recombination to take place, and hence a decrease in the number of charges. This decrease in number of charges will automatically give a decrease in current. And hence the baseline, the high baseline you had, will show a dip, will decrease, and this decrease constitutes the signal. So insecticides, pesticides, vinyl chloride and fluorocarbons can be very easily analyzed. So that's about detectors. Now the last one is a recorder, and then you have applications. So the sample injected here and you get various peaks. Peaks correspond to individual components. So applications of GC, you could have qualitative analysis or quantitative analysis. See each one of these. Qualitative analysis, it could be done for separation of mixture components. So the components can be separated and you get individual peaks, and these fragments can be collected separately. You can also do identification of compounds by retention time comparison. So if your standard gives a particular retention time and you get a peak at the same retention time, then you can identify that the same component is present in your sample. Pyrolysis, gas chromatography, um, this was about retention time. So in your sample, you are getting two peaks. This is the standard. So definitely this peak of the sample is representative of the same substance. So pyrolysis gas chromatography, it's used for the identification of non-volatile materials. I hope you remember this. This term I had used in our first class where I told you about modifications of GC. So plastic, natural and synthetic polymers, some microbial materials, these could be identified by pyrolysis gas chromatography. It's based on the fingerprint chromatogram for the sample, which results from its thermal dissociation and fragmentation. Basically, a high temperature is used. You break up these huge molecules into smaller fragments, and those fragments which are volatile are then separated by gas chromatography. Coming to quantitative analysis, you could have two ways of doing it, the external standard method and the internal standard method. First, I'll tell you about the external standard method. You prepare a set of concentrations. Here you have 5 nanogram per ml, 10, 25, 50, 75, and 100. Each of this will give you, each one is injected separately, each one gives a peak. When you compare, you will see that there is a progressive increase in the peak height. So peak height is plotted against concentration. You will get a calibration curve. You will get a straight line passing through the origin. Now you take the sample, you inject it, you will get a chromatogram. The peak height of the sample is then seen here on the graph. See where it intersects, find out the concentration. This is called the external standard method. You may be familiar with this. The internal standard method is slightly different. Here what is done is, to each of the standards, you are preparing the same standards, but to each of the standard, you will add same amount of a substance that elutes with a different retention time. So it could be an internal, that's called an internal standard. You watch here, when you are injecting 5 nanogram per ml sample, you will get the sample peak and you will get an internal standard peak. When you're injecting 10 nanogram per ml, again you will get a sample peak and internal standard peak. What you notice in all these standards is the sample peak, the left peak is increasing in height based on increase in concentration. But the internal standard peak remains the same. Why? Because in each of these standards, you will add the same amount or volume of internal standard. So same concentration of internal standard is present in all the standards. That's why it gives same response. The same is also added in the sample. So sample will give its own peak and this will give internal standard peak. Now, what you do is you will be plotting the peak height ratio. Ratio of the peak heights of these two peak heights, ratio you will plot here. on the y-axis, and x-axis is of course the concentration of your sample. You get again a calibration curve, and from the sample ratio you will take, you will see where it intersects the calibration curve, and then find out the concentration. So what is the use of internal standard method? The internal standard method is a more accurate analysis because there could be some variations in the response of the detector. Any variations in the response of the detector, or the way injection has happened, or anywhere in between, will be this variation effects will be eliminated because you are taking a ratio. Suppose, say, the detector responds more when you are injecting the sample, then it will show a larger rise for the sample. It will also show a larger rise for internal standard. The ratio will still be the same. That's why when you take the ratio of the two, it will eliminate the effects of any type of detector sensitivity changes. Aspects of DC applications. So it could be apart from qualitative, quantitative analysis, it could be used in food analysis. Analysis of food is concerned with confirming the presence and determination the quantities of analytes like lipids, proteins, carbohydrates, preservatives, flavors, colorants, and also vitamins, steroids, and pesticide residues. So those are things that are estimated in food analysis, can be used for drug analysis, widely applied to identification of active components, possible impurities, as well as metabolites. Environmental analysis, the environmental contaminants like dichloro, diphenyl, trichloroethane, DDT, and polychlorinated biphenyls, that's PCBs, are present in the environment at very low concentrations and are found among many of the other components. So DC, with its high sensitivity and high separating power, is mostly used in the analysis of environmental samples. Forensic analysis also, it can be used. Very little sample is available in these type of analysis, and the concentration of components is also very low. So GC is useful because of its high sensitivity and separation efficiency. Now this is something which I wanted to tell you. See, when you inject, this is the point of injection, you will always get a small peak in a gas chromatogram. This will be due to air which is not retained. So air will just come out because air is there. We are not doing in a vacuum the injection. So air gets injected. So this air peak will not be retained, and hence it comes out first. This is the time taken for an unretained solute to traverse the column, to pass through the column. Okay, this is required for certain calculations. This is your solvent peak, that is the carrier gas peak. Here you are getting two peaks. These are the sample peaks. Now this is the width of the first peak, and this is the width of the second peak. So what is the width of the peak? I'll give you the explanation of various terms in the next slide. I also mentioned in about peak fronting and peak tailing. Can you still hear me? Can you still hear me?
Yes, ma'am. Yes, ma'am.
Because I've had a power cut. I don't know whether I can still change this. Okay. These are the various terms that you have. Going back here, um, you have this is tailing. If you have a Gaussian shape, a proper shape, then you don't have tailing or fronting. If you have a peak that is getting extended here, that means it is tailing. If you have from this side, it will be called fronting.